Thank you, everyone. This has been great. It's such an honor to be here, and really appreciate the invitation. Let's see. Great. So I'm going to be talking about immunotherapy, and I'm going to kind of really talk about things at a very basic level, and just talk about the nuts and bolts of what we mean when we talk about immunotherapy, cancer immunotherapy. And there will be time at the end if people want to get into some of the nitty gritty about specific immunotherapies and specific sarcoma subtypes. So, you know, the things I want to really highlight in this talk are what are the most important things to know about the immune system related to immunotherapy? How does the immune system recognize cancer? And how can cancer immunotherapy help the immune system fight cancer? Oh. Next. Just the, okay. So a lot of patients will ask me, is this immunotherapy? So people take all kinds, people ask, they ask, you know, they have supplements. The supplement says on the bottle, it boosts your immune system. Why isn't that immunotherapy? And the, you know, well, first of all, we don't know if these things work, right? You know, so most of them, I don't think hurt. We certainly don't have any evidence that these supplements help. I mean, they're bad to take. I don't know. It's fine. I usually say it's fine, but that's not what I mean when I'm talking about cancer immunotherapy. So. There we go. So when I talk about cancer immunotherapy, I'm talking about a way to help the body specifically target the cancer. So, you know, you have all these different treatments. We have chemotherapy. We have tyrosine kinase inhibitors like cabozantinib and pazopanib. We've got molecularly targeted therapies like people were just talking about, using sirolimus to target the mTOR pathway, or using crizotinib to target ALK, right? Metabolic therapies, that's something that we don't have quite yet, but cancer uses sugar in a different way than other cells, and people are coming up with new treatments to specifically target that. But all these things are ways to distinguish cancer from the rest of the body. That's kind of the heart of cancer treatment, right? Chemotherapy targets the rapidly dividing cells. So immunotherapy is a way that we specifically target cancer and not target the healthy cells in the body. So how do we do that? So we're going to use the body's own way of recognizing things that are foreign. And the immune system, of course, is very complicated, but there's two basic cells in the body that are very good at distinguishing normal healthy cells versus other cells. And usually that's in the context of infection. So we need to be able to recognize bacteria if they're in the body. We need to be able to recognize virally infected cells. So B cells are a type of cell that are part of the immune system, and they make antibodies. So one way that the immune system recognizes something that shouldn't be there is with an antibody. The other type of cell that's very important, and is probably most important when it comes to cancer, is the T cell. And the T cell, as I'll show you in a second, can recognize things inside of the cell that shouldn't be there. So one thing I get asked about is, do we consider antibodies immunotherapy? And sometimes it's not a straightforward question, right? So Some people do, some people don't. I don't usually consider, if we're just administering an antibody to target the cancer, that's immunotherapy. We're still leveraging the immune system, but usually we're treating you with an antibody we've kind of co-opted the machinery. We've learned from the B cell how to make these antibodies, and now we're producing them in a factory and administering them to you. So I don't think that that's usually, I don't usually think that that's immunotherapy, because it's not your immune system that's being activated. But there's some debate about that. Some people would still consider it immunotherapy. So when I'm talking about that, it's, you know, in sarcoma, we don't have so many drugs like that, but one of the most famous drugs is Herceptin. That's a very popular drug for breast cancer, very important drug. It's a drug that specifically targets the breast cancer cells. It's an antibody. They make the antibody in the factory. They ship the antibody to the pharmacy. You, just like you're getting chemotherapy, you sit in the chair and they start to drip the antibody and it goes into your blood and starts to fight the cancer. And there really isn't a major one like that for sarcoma, but there are antibody treatments that are in clinical trials. There are some that might be promising. So these are things that are being developed. You know, does it matter if we call it an immunotherapy? Not really. It doesn't really matter what we call it, right? We want an effective treatment. But, you know, I'm here because I'm the immunotherapy expert. I'm going to be talking about immunotherapy. That's not what I'm going to focus on. Okay. What about T cells? How do T cells work? So T cells see things inside the cell. And when I was learning immunology, we didn't even talk about T cells in cancer. We talked about T cells and viruses. So viruses, that you get infected with a virus, but the virus doesn't just sort of float around in your blood. The virus finds your cells, maybe your cells in your lungs or your respiratory tract, any cells in your body, and it gets inside the cell. And it takes over the cells. It uses the cells to help it divide. The cells divide and it helps the virus divide. And that's how the virus works. It works inside the cell. So how could your body possibly find something that's hiding inside of the cell? It's very tricky, right? So your body has a mechanism, because viruses have been around for all of evolution. Your body has evolved to be able to see viruses. What your cells do is, every single protein that's in your cells are the body's constantly, it has a quality control mechanism where every single protein gets, a little bit of it gets taken, chopped up, and presented on the cell surface. And the T cells, they actually all have one specific problem that they're looking for, one specific alteration, and they're scouring the whole body. And every second you've got a bajillion T cells looking at a bajillion normal cells looking for a problem. And if they see a problem, they don't ask questions. They just kill the cell that's made a problem. Like that's their job. That's what we expect nothing less from our T cells. And they have a very high, I mean, you have to have very excellent quality to have a good functioning human body. That's right. So, but of course, okay. So and when viruses are inside the cell, they will have proteins that shouldn't be there, right? They have their viral proteins. And the T cells, that just like everything else, the body will scoop up some of those virus cells, hopefully, and present it to the cell on the cell surface, and the T cell will see that and they'll kill the cell. Now, of course, we know sometimes viruses can be very tricky and they can come up with ways to try and skirt these mechanisms. We're not going to talk about that, but this is the way things are supposed to function. So how do T cells distinguish cancer cell from the normal cells? Well, there's two types of proteins that the cancer can recognize. One are abnormally expressed proteins that are a normal part of the body. And what these are usually is proteins that are important for human development, but should not be present in adults. So this, if you see on your right, Yeah, there we go. Over here. This is actually the tube that brings sperm through the testicle where it needs to go. And the very, very, very primitive sperm are on the edge. And as they mature, they get closer and closer to the center. And everything brown is a stain for a very specific protein called MAGE4. And you can see that these cells at the very back, the primitive sperm cells, express it, but then as they get further and further along, they don't. So the only cells, this is actually the only cell in an adult, man or woman, that will express this protein. However, certain cancers will start to express this protein. And the immune system actually, because these cells have a bunch of different proteins in them, the immune system doesn't recognize this protein. It thinks it's foreign. When you have any place where the immune system is surveying, this protein shouldn't be there. And you can actually develop a response to this protein. So if any of you guys have heard about, we're going to talk a little bit in a second, some of these gene engineered therapies targeting NY-ESO-1, targeting MAGE4. These are targeting abnormally expressed immunogenic proteins that are found in the cancer. NY-ESO-1, MAGE4, they're present in the cells that become sperm. Sometimes some of these could be expressed in placenta. Some of them can be presented in certain fetal organs, but otherwise in normal functioning, healthy adults, they shouldn't be there. So the immune system recognizes them as foreign. The second one is mutated proteins. So as we were talking before, cancers have a mutated genome. That's how they became cancers usually, is because something in their genome mutated. So that mutation is not just sitting there in the DNA. That mutation actually is in a protein, and the protein is circulating in your cell. And just like everything else, that mutated protein can get presented on the cell surface. Now, we were talking before about tumor mutational burden. What does that mean? If you just have one mutation in a cancer cell, and some people do just have one mutation, sometimes one mutation is all you need to cause a cancer. But sometimes you have a cancer because that cell has lots of mutations. Lots of mutations. Each mutation, I mean, it's kind of like playing the odds. Each mutation has a chance of triggering a T cell to kill the cancer. If you have lots of mutations, your chances are much better that there's going to be a T cell out there that will recognize your cancer. And if you have a very high tumor mutational burden, that's a measurement of the number of mutations that are in your cancer, you have a high chance of there being T cells out there that are going to recognize the tumor. And that's why tumor mutation burden is a good biomarker, good measurement of whether a person might or might not respond to some of these immunotherapies. So how does some of the immunotherapies work? There's two basic ways an immunotherapy can work. One is you already have, your T cells are trying to fight the cancer, and they know where the cancer is, they see it, but they're just not doing a good enough job. The cancer is fighting back. We can get into how the cancer can do that. The cancer is fighting back. We need a treatment that's going to boost that immune response that's already there. The second way is that the immune system just doesn't see the cancer. It just doesn't know there's a cancer there. And we're going to redirect a functioning immune system, one that's ready to kill, to go to the cancer. So when I talk about strengthening the immune system, the immune response that's already there, that's checkpoint inhibitors. So that is PD-1 inhibitors, CTLA-4 inhibitors. What are PD-1 inhibitors? So the cancer has a number of different ways that it tells the T cells to go away or to turn off, or even though you see that I'm expressing, because people have problems with overactive immune systems. As you probably know somebody with problem like rheumatoid arthritis, lupus. These are all autoimmune diseases. If you let the immune system just do whatever it wants, I told you before how strict the immune system is. When the immune system sees something that it doesn't think should be there, it kills it, right? So sometimes the immune system could be overactive. It could be seeing a protein that's on the borderline. Your body has to also have mechanisms to kind of keep the immune system in check. So one of these things it has is called PD-L1. So the cancer will express a protein called PD-L1 on its cell surface. Here it's one of these blue arrows. And the T cell is ready to kill that cancer cell, but it has a protein called PD-1. And basically what the PD-L1, what it's really for is for a cell that you kind of know it's going to get killed by the T cell, and it has this flag that it can put up. It says, no, don't kill me. I know I look weird, but I'm supposed to be here. And that turns the T cell off. And the drugs that you guys are very familiar with, pembrolizumab, that's Keytruda, nivolumab, that's Opdivo, a bunch of other ones, avelumab, atezolizumab. All these drugs are aimed at blocking this junction that is the cancer cell turning off the T cell. And when you block that, in a lot of cancers, it works really well. The T cells just go ahead, and that was really the thing that was preventing the T cells from killing the cancer. And in some of the sarcoma subtypes, in undifferentiated pleomorphic sarcoma, a lot of the times, in alveolar soft part sarcoma, a lot of the times, that's all it takes for some of these patients. It kills it really well. Now, the other checkpoint that you may have heard about is ipilimumab or Yervoy, right? That's another very famous checkpoint inhibitor. That's another FDA-approved checkpoint inhibitor. So another one that's been tested in sarcoma. People have combined. So this one works a little bit differently. So instead of actually working on the cancer cell, your body has cells that are sort of in charge of telling the T cell which way to go. And these cells also have checkpoints, and Yervoy blocks over here. And usually these are the CD8 cells and these are the CD4 cells. So these are both very important cells in the immune system. And in some cancers, combining a CTLA-4 inhibitor and a PD-1 inhibitor works very well. It makes the treatments work much better. In sarcoma, maybe a little bit, but we're not really sure. Maybe a little bit. It also makes it a lot more toxic. So what are the toxicities of checkpoint inhibitors? Most people tolerate checkpoint inhibitors very well. I usually tell people that your chances are maybe about 50% that you have either no toxicity from the checkpoint inhibitor or very, very, very mild toxicity. Of the half of people that do have toxicity, it's still, for most people, very mild, but now it's maybe a mild rash, some fatigue that lasts a couple days after the infusion, some diarrhea that's well controlled with treatments from Walgreens. But there's a small percentage of people that could have real autoimmune problems. What do I mean by real autoimmune problems? For whatever reason, these drugs, they seem to activate the T cells that recognize endocrine organs a lot. I don't understand why. The thyroid is one that is frequently targeted. And it's not that uncommon for people on PD-1 inhibitors to have their thyroid. I mean, it's uncommon. It's less than 5% of patients, but we see it all the time. Patients who have their thyroid really messed up by the PD-1 inhibitor, even sometimes to the point where they need to take thyroid hormone for the rest of their life. Sometimes people have renal dysfunction, and usually that improves right away after we withdraw the immunotherapy, give them some steroids, and they kind of go back to doing what they were doing. Every once in a while, now I'm talking about a very unusual patient, like 1% of patients, people have very severe immune problems that wind them up in the hospital, like pancreatitis, like diarrhea so bad that they wind up in the hospital, like problems with their heart or their lungs. Any organ in your body can really theoretically have a severe autoimmune problem on the PD-1 inhibitor. So most people tolerate it very well, but there's a very rare section of patients who have very bad problems. We talked about tumor mutational burden. There's a new biomarker that I think is coming soon called tertiary lymphoid structures that people are going to be able to use to predict patients who don't have one of the common sarcoma subtypes who might respond to PD-1 inhibitors. So what about the other strategy I was talking about? What about redirecting your functional immune cells to fight the cancer? And this is happening right now. You've probably heard about some of these trials in synovial sarcoma, in myxoid round cell liposarcoma, where they're genetically engineering T cells to recognize the cancer. And it's a whole big process. You have to get a special kind of blood draw that requires you to sit in like a special chair hooked up to a machine. It kind of looks like a dialysis machine. It filters out your white blood cells because they have to take so many white blood cells that you couldn't just get it on a regular blood draw. It like filters them out and then gives you back your regular blood. They take those white blood cells to a special processing facility. They genetically engineer them, but they can't, it's more complicated than that because they have to genetically engineer a small number, and then they have to grow that small number out into many, many hundreds of billions of cells to get back to you. And then you get a high dose of chemotherapy. So it's not easy, right? It's intensive chemotherapy that you have to get. And the reason why they do that is not to kill the cancer with the chemotherapy. It's actually to kill your immune system. So they kill your immune system so that your body starts producing all of the growth factors and signals that say, hey, we need to grow immune cells. And then your body's producing that as you get this infusion of T cells, and it helps those T cells grow and take root and expand and fight your cancer. And so right now there are trials in synovial sarcoma and myxoid round cell liposarcoma. Those are using TCR engineered T cells. So those are just basically what they've done is they've taken T cells from a patient who was able to kill their cancer with T cells, and they copied that T cell receptor, and now they're putting that into healthy, or into your T cells that don't have that T cell receptor so that it can kill the cancer just like theirs did. There are other receptors, CAR T cells, CAR receptors, if you've heard about this. These hook up the internal machinery of a T cell with an antibody. So the antibody can actually see things on the cell surface. And that has advantages and disadvantages. The CAR T cells have been tremendously effective in certain types of leukemia. They've totally changed it. Patients who just a few years ago would have had uniformly fatal leukemia can be cured. But we're still kind of at the beginnings in sarcoma. There are some trials looking at this sort of drug and looking at this sort of cell product in sarcoma, especially with some bone sarcomas, focusing more on the pediatric types of bone sarcomas. But I think they will get to everybody eventually, or everybody with the target. So what are some other strategies? Well, so we talked about the checkpoint inhibitors, but these are just a section of the checkpoint inhibitors. There's actually many checkpoints in your body. There's really two that we talked about. There's another one where there's an FDA-approved agent now called LAG-3. There are a bunch of other checkpoints that people have drugs for, OX40, IDO, a bunch of them. And I think some of them have had missteps in terms of the drug development, but I think some more of them will move forward. Cancer is not just cancer. The tumors are not just cancer cells. The tumors are often, they may only be 50% tumor cells a lot of the times, maybe 50% tumor cells, and then 20% your T cells that are futilely trying to kill the cancer cells, and 10% inhibitory cells, and and so on. So it may not be the cancer itself that's able to deflect the T cell response. We don't see a lot of regulatory T cells, but that's one type of inhibitory cell that can be in the cancer. More common is a certain type of cell called a macrophage. And macrophages are, a lot of the times I think of them as being kind of the cleanup crew cell. Like, so if you have cells that are dead anywhere in your body, like if you have a burn or something like that, a wound, your body doesn't want to just let the dead cells sit around. You have to get rid of them somehow. So these cells called macrophages, they come in, they eat up the debris, and they present it to the T cells. And sometimes if there's like a necrotic infection, right? Like if there's like a bacteria like a flesh-eating bacteria, those cells can be very important because they could go into the debris area, eat some of those bacteria, and show them to your immune system and say, hey, there's a problem here. You have flesh-eating bacteria. You got to send arsenal of immune cells down to this area and take care of it, right? But what can also happen is if you have a burn, you may not want a huge inflammatory reaction in the burn. You want to heal the burn, right? So you want to say, hey, that's what you're going to put some antibiotic ointment on the burn, right? Because you don't want it to get infected. Your body is going to not focus on fighting any potential infections. Your body is going to just focus on healing the wound. So actually, these cells they can have two modalities. They can say, hey, bring the inflammation right over here to fight an infection. Or they can say, hey, go away, we're focused on wound healing right now. And a lot of times in the cancer, we know that they have all of the properties of telling the immune system, go away, there's a wound here. So your body thinks the cancer is a wound. We think that in a lot of sarcomas, that's the main mechanism of immune evasion, especially leiomyosarcoma. There's really good evidence that that's what's going on in leiomyosarcoma. And there are so the very common drug trabectedin that I bet a lot of you guys have been on, that is one of the things that trabectedin does, in addition to killing the cancer cells itself, the trabectedin really kills those macrophages in the tumor. And actually we had a trial, a bunch of people have been interested in combining trabectedin with immunotherapy to try and have this kind of synergistic effect. And I think it's, there's something there to it, but I don't think it's panned out as well as some people hoped, like a lot of things. But I think there's a lot of really promising strategies for how to kind of convert these macrophages to make them kill the cancer or create an immune response against the cancer rather than tell the immune system to go away. Vaccines are something that I'm always a big fan of. Vaccines are, instead of redirecting the cancer by redirecting your T cells by engineering your T cells, you have T cells that should be fighting the cancer. Why not just boost them with a vaccine? So that's something that we've tried before. I thought we had some promising data. It wasn't quite good enough to make it, but there's some new vaccines out, and I think there's going to be even more in the coming years. So I still believe in cancer vaccines. I think that they're gonna. I think we're going to figure it out. Natural killer cells are another sort of kind of T cell that can work in tumors that aren't presenting their mutations to the T cells very well. Cytokines are basically your body's signals to activate the T cells. And back in the day, people treated patients with just by purifying those cytokines and giving them to patients. And actually in melanoma, they worked pretty well, not compared to the new drugs, but back in the bad old days, they worked pretty well. And there's a bunch of companies now that are trying to basically study the cytokines and engineer them to make them work better than normal. And I think some of them look pretty good. And I think some of those are gonna be in clinical trials for sarcoma in the coming couple of years. And then chemotherapy and radiation also have immunogenic effects. So just by killing, if we can time things right between the immunotherapy and the chemotherapy, maybe we can kill the cancer and also stimulate the immune response as well. So, so we talked about the most important things to know about the immune system. We talked about B cells and T cells. We talked about how the immune system recognizes cancer, right? We talked about antibodies, and we talked about T cells and how the T cells can recognize either abnormally expressed proteins or mutated proteins. And then we talked about how immunotherapy works. It works by either redirecting the immune system to recognize the cancer, or by taking the T cells that already recognize the cancer but are being inhibited somehow, possibly by a checkpoint. So with that, I want to say thank you, and I'll take any questions. Thanks.
Thank you so much. Any questions for the audience here? We're seeing if there's some in the chat right now as well. I'll be right there.
I was curious because of chemotherapy and the immunotherapy. Is there like, would you do the immunotherapy first and then like a chemo, or is there a certain order that would kind of coincide, or at the same time? Even?
Well, so in terms of combinations of chemotherapy and immunotherapy, those are generally being done in clinical trials. So there are a number of clinical trials going on around the country, and there's going to be more in the coming years. There's one at the University of Colorado that's open right now. I think there's one multi-center trial that's going to open a cohort that should be available like around the country with chemotherapy and immunotherapy combination. There are a number of small trials kind of around the country looking at this. There's a number of trials that I think probably are still a little bit earlier in development that are going to be widely available. So that's kind of, you know, it's coming. This is kind of like the cutting edge. Now, in terms of should you get immunotherapy first or should you get chemotherapy first, you know, the way this is more of like just a standard drug development question, right? So we usually either you're getting the standard of care first, or you're going to get a clinical trial that's kind of like well designed to kind of integrate into the standard of care. So for example, we had a trial of doxorubicin and a PD-1 inhibitor. So that a lot of people got up front, right? Just when they would normally get doxorubicin. Whereas if you are trying like a very new experimental phase 1 immunotherapy trial, usually those aren't for people who've never had any chemotherapy before. Usually those are for people who have kind of tried the standard drugs and they didn't work. Yeah. Yes?
I actually have a couple of questions.
Wait.
Oh, I'm sorry, Charles. No.
Hi, one was that there was a recent report that sarcoma cells actually produce a protein, MIF, that affects the immune system in some way and keeps them from attacking the cells. Is that something that could become a targeted treatment?
Yeah. Yeah, there are a few of those, right? There's, I know what you're talking about. There's another one called TGF-beta. That's a one that I think is very important for a lot of sarcomas. It's tricky to figure out how to target these things because just like everything else, when you talk about the immune system, these things are there for a reason, right? They're part of your normal healthy immune response, and your, you know, there's potential for autoimmunity if you turn it off too much. But yeah, no, I think that these are all, that's definitely kind of the type of thing that we're working to target.
And the second question is maybe not immunotherapy per se, but there are, nobody's talked about like oncolytic viruses.
Oh yeah.
And I'm wondering does the immune system attack the cell with the viruses in it, or?
Yeah, actually that's probably the, you know, people think that, okay, we've got an oncolytic virus, we've engineered a virus to go and kill all the cancer cells, but actually it's really nobody's made a virus that good yet. And most of the kind of success that you've seen with oncolytic viruses has actually been by triggering the immune response against the cancer. Usually what happens, the way those things work. And so there's one FDA approved oncolytic virus called TVEC, and that has been tested in a couple trials of sarcoma. And the TVEC is kind of, it's FDA approved. So I don't like to go around saying things that are FDA approved aren't that good, but it's, you know, it's an incremental step, right? It's kind of like it doesn't, you know, when people talk about oncolytic viruses, the idea is that you're going to engineer a super smart virus that can just kill the cancer and spare your healthy cells. And TVEC is not really that, right? And but there are people that are working on that, right? And they range from anywhere from TVEC plus just a tiny bit of efficacy all the way to, you know, really complicated smart viruses that have their own set of problems, right? But yeah, so right now, TVEC, the way that works, the way most of the oncolytic viruses on that part of the spectrum work, are that the cancer cells just fight the virus less well than the healthy cells. And when the immune system recognizes there's a viral infection there, they tell all their friends to come and check out what's going on here. And that just leads to a better cancer T cell response. That's probably the main way that the oncolytic viruses that are in clinical trials right now are working. Yeah.
We have a couple of questions from the folks watching us via Zoom, and then Brandy, I'll get to you right after that.
So one of the questions online was, if on immunotherapy treatment, your lymphocytes start to decrease in number, say to mild lymphopenia, is that of concern? And what does this mean for treatment?
Yeah, no, that's a good question. So it's, I think it probably is a problem for people like over the many years, like their cancer takes a beating and their immune system's taking a beating. And I do think that over time, it's sort of an inevitable and unavoidable problem that I do think there's some sort of weakening of the immune system that, you know, that helps the cancer be free. And I don't know what to do about that, because you have to keep fighting the cancer. I think probably what I would like to do about it is move some of these immunotherapies further and further up front in treatment. And we are trying to do that. We are trying to figure out how to do that. But how do you do that without sacrificing the benefit of giving the chemotherapy early? That's really what we're trying to figure out. Now, in terms of like just on like a person's, like during the time of a person's chemotherapy cycle, they're going to go through a time of leukopenia, and then their white blood cells are going to rebound. I don't think that sort of short window of time where you're immunosuppressed is going to make a difference in terms of the cancer immunotherapy.
Thank you.
Mm-hmm.
And what are your thoughts on antibody drug conjugates in metastatic sarcoma?
Yeah, so yeah, this is, I think antibody drug conjugates are really cool. I think that what an antibody drug conjugate is so this kind of at the beginning we were talking about, are antibodies immunotherapy? You know, I'm not sure it's not what I'm into, but it's not, you know, I don't know that it matters, right? They're an important part of the future of cancer therapy. So antibody drug conjugate works. You have an antibody just like you have antibodies for COVID or, you know, whatever, right? The antibody is something that your B cells make to recognize something foreign, and they've made it to recognize something on the cancer. And a lot of times I was talking about Herceptin, right? That's a drug for breast cancer, targets HER2. The antibody is enough. The antibody will kill the cancer just on its own, or it'll mark the cancer to be killed by your body. Antibody drug conjugate is an antibody that's been linked to another drug. So it's the antibody recognizes the cancer, but it carries a payload. And a lot of times that payload is a poison. So not only, maybe the antibody on its own is not strong enough to kill the cancer, or maybe it will do a little bit of damage, but not enough to write home about. But the antibody drug conjugate carries a poison. And for whatever reason, when it gets into the tumor, it releases the poison. And they've got all kinds of fancy chemistry using a bunch of different mechanisms to make the antibody release the poison. For example, the tumor may be more acidic than the rest of your body. So when the antibody drug conjugate is circulating in your blood, it doesn't release the poison. But when it gets into the acidic tumor, the chemical bond between the antibody and the poison gets broken, and then the poison gets released into the tumor. So I think that there are a number of antibody drug conjugates that are in clinical trials right now for sarcoma that I do think are very promising, that I do get excited about. They're early in their development, but I think this is something that, it's something that, people who are following what's kind of coming along through the pipeline, it's exciting. Yeah. Hey.
Hi, yes. Who are not considered as good candidates for immunotherapy? I was not given the option at all. It was either chemotherapy or radiation. And if none of those work, it was amputation.
Yeah. Well, so right now, in terms of standard of care, there's nobody with sarcoma that's getting immunotherapy as part of curative treatment. Most people getting immunotherapy are getting it in the metastatic setting when they have incurable disease. Now, there's a major national clinical trial, the SARC-32 study, that has been testing the addition of immunotherapy to neoadjuvant radiation in patients with undifferentiated pleomorphic sarcoma and dedifferentiated liposarcoma. It's a randomized trial. So if that is successful, if that's positive, then I think people will start to receive immunotherapy as part of their initial curative therapy, as they're getting in a lot of other cancers. So there's a lot of other cancers where people are getting immunotherapy, and it makes a big difference in terms of improving their chance of cure. But yeah,
but why wouldn't it just, you kind of assume that it would be okay to start it? Because if we have T cells and that's what it's activating and enhancing that, why wouldn't that?
Well, yeah, so I agree, but so it kind of goes back to the clinical trials discussion. Why don't we, am I going to get a placebo? Am I going to get if I'm getting a clinical trial, does that mean I'm giving up on my real proven treatment? We have to do things in kind of I feel the same way. I think let's just give everybody immunotherapy right at the beginning, right? That's kind of like what my gut says, right? But I've been wrong before, right? So, we have to do things in a organized way, right? So, we started with immunotherapy in the metastatic setting, right? And we kind of got a sense about where it's most effective. And now it's moving to the curative setting, and it's being tested in a careful way. And I think we'll probably know, maybe even in a year or two, we may have the first data from that trial. Yeah. Mm-hmm. Yep.
Well, thank you so much for that. I mean, immunotherapy is probably the hardest topic to talk about. And I was listening very closely, because when